In this session, we will discuss T and B cells differences and their main role. Let's make comparison of T and B cells. Because these are important points, you need to remember about the T cells and B cells. Origin of T cell is bone marrow. Origin of B cell is also bone marrow. T cell matures in thymus, and maturation of B cell occurs in bone marrow. Peripheral blood has 70 to 80% of total T lymphocytes. Peripheral blood contains 10 to 15% of total B lymphocytes. CD markers of T cells are 3, 4, 8 that are involved in cell-mediated immunity. Cell-mediated immunity is carried out by T cells or T lymphocytes. In cell-mediated immunity, T cells help B cells in producing antibodies, or they may go ahead and destroy the pathogen itself. CD markers of B cell are 19, 21, and 24, and involved in humoral immunity. Humoral immunity is carried out by B cells or B lymphocytes. The response of B cells is that antibodies are produced in the blood in response to the entry of pathogen. T cells are of 4 types, which are helper T cell, memory T cell, cytotoxic T cell, and suppressor T cell. You can remember these by a mnemonic. Help me catch some. There are 2 types of B cells, and that are plasma cells and memory cells. You can remember it by a mnemonic. My pleasure. As we have discussed in our previous videos that CD8 binds with MHC1, while CD4 bind with the MHC2. But how to remember it? Well, remember MHC1 versus MHC2 through the rule of 8. Let me explain it to you. 2 into 4 equals 2, 8. Similarly, 1 into 8 equals 2, 8. Before ending our video, let me ask a question to you for your self-assessment. If T and B lymphocytes both are produced from common lymphoid precursor, then why T and B cells are different from each other? Are you able to answer it? Let me answer it, and then compare our answers. Inside the bone marrow, there are stem cells. These are called pluripotent stem cells. In the production of lymphocytes, some of the pluripotential stem cells become early lymphocyte progenitor and then lymphopoietic stem cells, which are also called common lymphoid progenitors. To become B cell, it differentiate into immature B cell in bone marrow. But let's move into depth at how lymphopoiesis of B cells are different from T cell. As both have common lymphoid precursor in common, in case of B cell, common progenitor precursor remain in bone marrow until mature. So the differentiation of B lymphocytes in the bone marrow can be followed by looking at the expression of different molecules by the developing B cell. One of the first signs that a cell is committed to the B cell lineage is the expression of a surface protein called CD19. The cell at this stage is called a pro-B cell. The cell then expresses the IgM heavy chain in its cytoplasm and is known as a pre-B cell. The next stage is called an immature B cell, which expresses immunoglobulin M on its cell surface. Finally, the cell becomes a mature B cell, expressing both immunoglobulin M and immunoglobulin D on its surface. This B cell leaves the bone marrow and enters the bloodstream. But in case of T lymphocytes, after formation of common lymphocyte progenitors that will move to thymus and forms thymocyte and then complete its development as mature T cell. The mature T cell having gamma and delta will be expressed as gamma and delta T cell receptor. But T cell having alpha and beta will be expressed either CD4 or CD8. Then they are released in blood steam. That was all about today's video on primary lymphoid organ and lymphopoiesis. Stay tuned to sqadia.com for more precise and knowledge-stuffed videos.